Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So here we will discuss about the microbiota that is play important role for absorption of waste foods by starch fermentation to produce short chain fatty acid. So in our gut, large intestine contain a bacteria. That is bacteria play important role as a mutualism, and that's a type of symbiosis. It means the bacteria is also helpful for getting food while it's produce short chain fatty acid, which that is the good for our health. So that is the microbiota are present in large intestine, but there are many bacteria is harmful in our gut, which that is resist by the mucus are present in the mucous membrane in the large intestine. We will discuss here. So here is the person which contain a digestive system. Digestive system is made up of esophagus, stomach, liver, pancreas, small intestine, large intestine, and that is basically large intestine contain food which that is wasteful. We will discuss and get a more concept. So here is the different type of grains diet which that we are used, that is made of the amylose or amylopectin. So the amylopectin 1 to 6 beta glycosidic bond, which that is in the form of branches, while the amylose alpha 1 to 6 glycosidic bond in the linear chain. And on the other hand, there are many sugar molecule which that is the monomer of the sugar, while the oligosaccharide or disaccharide is also present. So that's known as the starch, amylose and amylopectin. When we will eat the starch, that will be partially digest in our oral cavity. That's oral cavity contain a amylase enzyme, which that are present in the parotid gland and some mandibular gland and sublingual gland in our oral cavity. So here I am drawing the large intestine. Large intestine is begin from the ileum of end of the ileum. Ileum is the small intestine, end of the small intestine, and ileum allow to move the sugars or carbs inside. So there are different type of accessible carbohydrates in the microbiota. So the microbiota will be used. So here is the ascending, descending, and transverse colon as well as sigmoid colon, and here is the rectum and anus, and that is the appendix. So that is the basically is a part of large intestine. So the large intestine, the function is beside the microbiota, that is the absorption of the salt and absorption of the some nutrients as well as the absorption of the vitamin and water. So that is the important character for the large intestine, as well as the expelling of the feces. So here is the food when intake into the stomach, that's converted into the bolus, and this bolus is after the mixing of the enzyme into the stomach to go in the form of the chyme. So this chyme is basically simple convert into the simple sugar by the activity of pancreatic amylase enzyme, and sucrase and disaccharase enzyme are present in the enterocyte. So here is the blood after the conversion of simple sugar which go into the blood. So however, that means the when the waste carbohydrate which that is undigestible in our small intestine and more in the large intestine, so that is the go into the large intestine. So what will be happen in the large intestine, here we will discuss. So this is the cecum. This cecum contain basically the microbiome which that begin to the large intestine. So first of all, I want to say that the harmful bacteria are present also, and some bacteroids. So these bacteria is created by the evolutionary process in our large intestine to create a barrier. So that's known as the barrier immunity. While here is the esophagus, stomach, and duodenum contain a pyloric sphincter, and that's the beginning of the stomach known as the cardiac sphincter. So that here is the duodenum which contain a mucocyte as well as enterocyte. On the other hand, there are enteroendocrine cells also present in this way. And this is the wall inside the lumen contain bacteria. It's the slightly amount of bacteria which that is due to the acidic medium in our stomach. So that means here is the large intestine which contain a harmful bacteria as well as helpful bacteria to create a microbiome. So this bacteria is not penetrate into the enteroendocrine enterocyte, while the microbiota is live into the lumen, and that is not go due to the mucous membrane production from the mucocyte. So here is another important thing, that is the barrier immunity is the mucous membrane, while here is the macrophage and dendritic cell is also known as the Langerhans cell, which that is the innate immunity which will be allowed to engulf the pathogen to prevention of that pathogenicity. So the remembering thing, that is the barrier immunity, and further go into the innate immunity. After that innate immunity, the memory cell will produce the antibodies known as the adaptive immunity. So here I am giving the cross section of the basically inside of the lumen which contain a microbiota, and this microbiota accessible carbohydrates will be occur. So these accessible carbohydrate, here is the enterocyte and here is the blood vessel. So the accessible carbohydrate can be fructooligosaccharides, and cellulose and resistant starch. So the resistant starch means the resist to the digest in our gut. So that is difficult to digest in our gut. So how to break down it? It is the help by the it is through the bacterial interaction to use as the energy to produce energy by the bacteria as well as produce the short chain fatty acid, which that is the helpful for the human body. So remember, that is the microbiota in our gut in the lumen inside. So here is the bacteria divide more and more to maintain their life inside, to maintain their population. So here is the fructooligosaccharides is a type of resistant starch. So that is the accessible for the bacteria. On the other hand, cellulose. Cellulose is the part of used for the cellulase enzyme to break down into the simple sugar. So the Rheuminococcus bacteria is used for it, while the resistant starch is basically the unable to digest with our own enzyme. So the bacteria is useful. So the primary degraders is first of all Bifidobacterium species and bacteroids and Rheuminococcus. So that some bacteroids is harmful in our gut. So some bacteroids is useful. So this Rheuminococcus, and this produce the different type of short chain fatty acid is known as the propionate, formate, and acetate. This propionate and formate as well as acetate is cross feeding. That's cross feeding means the another bacteria used for it to cross feeding to produce again acetate also, but also methanogen, which that is the bacteria methanogen produce the methane gas, while the acetogen is also present in our lumen, which that is used for the cross feeding to produce again acetate from the cross feeding from formate or propionate to again produce acetate. That is the conversion of the molecule of the short chain fatty acid. So here is the glucose also produced, and this glucose is separation by the primary degraders. So these primary degraders will be converted into the secondary degraders used, that is the firmicutous species, which that's converted into the butyrate, and that is the basically butyrate is the short chain fatty acid. So the short chain fatty acid is play important role for the anti antioxidant as well as quick energy source for brain as well as heart, etc. And that is also the composition of the vitamin, and bacteria also produce the vitamin K as well as the vitamin A. So that is the type of omega 3 fatty acid, and omega 6 fatty acid is also produced from it. So that is the short chain fatty acids contain a carboxylic group. So that's ultimately go into the blood, and where the need of these it provide, and these blood our blood will be provide the short chain fatty acid in our tissue as well as use as a energy. So here the microbiome modification in gut is due to the diet, exercise, as well as age. So the age related the different type of bacteria, which that is a helpful is age will be matter. For example, number of bacteria and the quality of bacteria is in the form of different types. So these different types is increase or decrease by the changing of age. So I can explain about this diagram to understand as well. So here is the horizontal show that the age, and the vertical shows that the quality of different bacteria colony or different bacteria species. So you can understand as well. While on the other hand, the genetics is also matter. For example, all bacteria is about human infection resistant by gene expression in the large intestine. So the large intestine contain DNA in the nucleus, which that express to produce mucopolysaccharide as well as mucous membrane, which that is barrier immunity to allow prevention. It is not allowed to move or invade inside of the bacteria, inside of the enterocyte, while this is the DNA expression in the form of RNA, that is known as the transcription, and that's transcription again. RNA will be produced the protein by using of transfer RNA is known as the translation to produce protein. That's protein in the form of mucus producing genes, immunological response, acidity, antibody like protein expression. So that's all thing is due to the microbiota, which that is the our bodies adapt to resist